So now we have to explain the mechanisms of hormone action, how it affects the target cell. So different cells have different numbers of hormone receptors, and an increase in these receptors would be an upregulation that makes the cell more sensitive to a hormone, and a downregulation would be the decrease in the receptors, making the target cell much less sensitive, which is what happens in type 2 diabetes. The receptors for the insulin is decreased, so you're not really sensitive to insulin anymore. So there are actually kind of like two systems. I'm just gonna explain the more easier to understand. Well, not really, but anyways, we'll just explain the intracellular receptor and a direct gene activation. So there are two hormones that does this. One is the steroid hormone because it is lipid soluble. It's lipid derived, so it can just enter the plasma membrane, which is also lipid soluble. It's made of lipid, like right in the middle, like the hydrophobic tails. That's made of lipid, and it just goes straight through. The other hormone is thyroid, which people thought was lipid derived, but it just looks so efficient that in actuality, it's actually using a transport protein. So these hormones get into your cytoplasm, attach to a receptor complex. This complex then go through the nuclear plasma, which is the nucleoplasma, and affects DNA in a specific area. There's a specific name that I'll put up in the information bar. I'll put all this process on information bar because I'm probably talking a little bit fast. Don't know if you can understand me, but goes to the DNA. DNA changes the translation rates, and there's a special thing about thyroid hormone being able to not only affect the nucleus but also the mitochondria, and that increases your basal metabolic rate. So the other system is called a second messenger system. In that one, the hormone never reaches the cytoplasma. It only attaches to the receptor on the plasma membrane, and then the receptor activates the G protein, the GS protein to be specific, S standing for stimulating. So this G protein is involved in all second messenger systems, and is composed of multiple parts, and it's heterotrimeric in structure. So there's a beta alpha dimer, a alpha unit, which has the GDP, and when it's activated, the GDP comes along, the GDP goes off, the GTP enables the alpha unit to dissociate from the beta alpha dimer, and it moves over to the adenylate cyclase, which turns ATP into cyclic AMP, which then affects the protein kinase A, which then does all kinds of enzymatic activities, processes. So the second messenger is actually the cyclic AMP, and you don't want to have it remaining in the cell forever. So there's a phosphodiesterase, PDE, that comes along and kind of cuts down the cyclic AMP, and there you have the second messenger system. So the other system that's also being mentioned is the PIP calcium signal mechanism. Oh, before we go on to that, I have to say that the hormones that use the second messenger, there's a variety of them. There's the ACTH, the catecholamines, the glucagon, and various others. And in a second messenger system, the hormones can be very effective at low levels because they have an amplifying effect. You only need like 1 or 2 hormones to get a lot of cyclic AMPs, and that'll activate a lot of protein kinase A, and it just kind of like balloons. So, going on to the PTP, calcium signal mechanism, that does the signal transduction. It also has a G protein, but instead of a adenylate cyclase, it uses a phospholipase C, and it that turns the PTP2 to DAG and IP3. DAG then activates PKA, so DAG is kind of like cyclic AMP, and then that's activated, and the IP3 goes over to the endoplasmic reticulum, the ER, to release calcium. Calcium then like gets itself like to a calmodulin, and that affects like the intracellular processes. So there you have the mechanism of hormone action. I'll be sure to include information that I forgot about in the information bar.